Welcome everyone to today's webinar with Dr. Seth Lorenzino from Intellia Therapeutics. She's the Chief Scientific Officer and Vice President there. And she's going to tell us everything about CRISPR-Cas9 gene editing, the Intellia way. As usual, everyone will be muted. Please put your questions into the chat pod and Dr. Seth Lorenzino will get around to answering them at the end. Over to you.
All right, thank you. So hello everybody who joined, thank you for doing so, and thank you Petra and OTS for the opportunity to tell you about CRISPR-Cas9 and how we're using it at Intellia to make human therapeutics. So before I need to share with you the forward-looking statement, but this moving on to the agenda, I'm going to be covering 4 topics. First, I'm going to give you an introduction to Intellia's modular genome editing approach. Then I'll give an overview of CRISPR-Cas9 and how we're applying it. And then in the third and fourth part of the talk, there will be examples of how we're deploying these to make human therapeutics for an in vivo knockout approach, as well as an in vivo insertion approach. So this slide has the company's mission, right, which is to develop curative genome editing therapies. But I like to think of it as what's the vision? And the vision is a world without genetic diseases. And that's a really exciting vision that keeps, me and my colleagues really very engaged and dedicated, to make this happen. So central to this vision is the use of CRISPR-Cas9 as a tool to specifically and precisely introduce edits in the human genome. And these nucleic acid changes can lead to inactivating or deleting a disease-causing DNA sequence through a knockout approach, or to correct an error, a misspelled DNA sequence through a repair mechanism, or to insert a new DNA sequence or a sequence that has a new functionality for cases of loss of function. So at Intellia, we're leveraging a modular platform of both gene editing tools like CRISPR-Cas9, as well as delivery solutions to advance a pipeline of differentiated candidates that allows us to go after genetic diseases. And the way we're deploying this platform, this modular platform, is in vivo. We're using CRISPR as the therapy. And here, the first organ we're targeting is liver, and I'll tell you more about that. And the goal is to go after genetic diseases. And then on the right side here, we're using CRISPR as a tool for us to be able to introduce defined changes, and in the majority of the cases, several changes into a cell to rewire it with new instructions. So the cell will be a living drug after we reintroduce it into the patient. The ex vivo approach, as you'll see next, we're using it in the clinic for hematopoietic stem cells for sickle cell disease, and we're developing candidates for immuno-oncology and autoimmune diseases. Today, I'm going to focus my talk on the in vivo side of the business, and hopefully in the future I can come back and tell you more about what we're doing ex vivo. So here is a snapshot of our pipeline. So in red is the in vivo pipeline where CRISPR is the therapy. So here we have NTLA-2001, which is our first systemic CRISPR drug candidate, and this is the first ever systemically administered CRISPR candidate. This is in the clinic for transthyretin amyloidosis. And this is in partnership with Regeneron, with Intellia being the lead party. We have a second candidate called NTLA-2002 for hereditary angioedema. And then we have a number of programs in the research stage. A couple of them are insertion for hemophilia A and B, also in collaboration with Regeneron, and a number of knockout, insertion, and consecutive edits that are fully owned. On the ex vivo side, as I just told you, we're in the clinic with our partner Novartis for sickle cell disease by editing hematopoietic stem cells. And then we have a pipeline of wholly owned autoimmunity and immuno-oncology candidates. The first one is Intellia 5001, which is an autologous cell therapy in which we have a T cell receptor that targets a cancer antigen called Wilms tumor 1. And this is a candidate that we're going to be moving into clinical development next year. So what is CRISPR and where does it come from? So, CRISPR is an abbreviation for cluster regularly interspaced short palindromic repeats. And they were first identified in bacteria, in prokaryotes, in bacteria and archaea. And what the investigators realized is that there were these CRISPR arrays, that there were repetitive sequences that contain spacers that were clustered in this array. And what they found is that these spacer sequences, they match the sequences of bacteriophages that could infect those bacteria. So there were a number of really elegant studies that demonstrated that these CRISPR arrays and associated proteins called CRISPR-associated proteins, or Cas, played a role in an adaptive immune system in prokaryotes. And the way this system works is that when a phage infects a bacteria, the spacer sequences in the CRISPR array are transcribed. They generate short CRISPR RNAs. These bind to the Cas proteins, and they recognize any inviting viruses like bacteriophages. Once that they recognize that virus, by sequence complementarity, the Cas protein cuts the viral DNA and prevents reinfection of that bacteria. So, a really simple and effective way that depends on an endonuclease called Cas and a guide RNA that will tell that Cas where to make a precise nucleic acid cut. So the potential for CRISPR to really precisely manipulate the genome really became apparent to many researchers, and including the founders of Intellia Therapeutics, and they demonstrated how to repurpose this prokaryote system for the manipulation of eukaryotic genomes. And this is any genomes. There is a lot of work being done on plants, and what I'll tell you today is what we're doing for therapeutic use. So the modularity from the system comes that you have a constant enzyme, which is the Cas enzyme, that can be reprogrammed for identification and introducing a DNA cut on a specific target just by changing the guide RNA that's going to bind to it. And these, although there are other mechanisms for introducing DNA changes, zinc fingers, meganucleases, TALENs, etc., what differentiates CRISPR is that the only thing you need to change from target site to target site is the guide RNA. And as we will see in the next slide, it's just a little piece of the sequence of the guide RNA that needs to be changed. So genome editing by CRISPR-Cas9 is efficient, is simple, and is scalable. And that's what really has led to this revolutionary use of CRISPR-Cas9. And there's thousands and thousands of papers being published and new applications of this technology. So going a bit deeper on the mechanism on how CRISPR-Cas9 can be used to generate precise gene modifications. So there are a number of factors that contribute to the specificity. First, it's important to know that Cas9 is inactive when it is not bound to DNA. So it's a programmable endonuclease that will only become activated after the guide RNA binds to it. The guide RNA shown in teal, it has a scaffold region that is the main area for interaction, domain for interaction with Cas9. Once that it binds, it forms a ribonuclear protein complex. The other important region of the guide RNA is, in the case of Streptococcus pyogenes, is this 20 nucleotide region on the 5' end of the guide RNA that determines the target site by Watson-Crick sequence complementarity. So the recognition of the site is dependent on two things. One is this 20 nucleotide stretch, but also importantly is a domain that is on the complementary strand that's called the PAM domain, or the protospacer adjacent domain, shown in the cartoon in yellow. So the Cas9 with the guide RNA loaded onto it scans the DNA, identifies the PAM, and then the target site hybridizes to the complementary sequence. There are a number of conformational changes that happens through all of these steps, which eventually results in the activation of two nuclease domains in the case of SP-Cas9, the HNH and the RAPC domain, that lead to a double strand break on each strand of the DNA on the target site. So what happens now is that you have this double strand break that's recognized by the endogenous DNA repair mechanisms of the cell. And there are two main mechanisms. On the left, it's the mechanism called non-homologous end-joining, or NHEJ. This is operational in both proliferating as well as quiescent cells. So this is the default mechanism. This is an error-prone mechanism, so the cat is going to be rejoined, not perfectly, so they could be small insertions and deletions. So this is called the indels. And that results in mutations in that target site that will lead to disruption of a coding region, for example, or to regulatory elements or to binding elements that will disable the gene. On the right side is another mechanism called homology-directed repair, or HDR. This is only operational on actively dividing cells. And it depends on the presence of a donor template DNA that could be a single-stranded oligonucleotide or a plasmid or other forms of DNA that has homology arms, and through homology-directed repair, you could correct a single nucleotide error as well as insert a larger stretch of DNA in a knock-in approach. So now, having covered the mechanism, how do we use this technology to identify guide RNAs that are suitable for human use? And here I really want to highlight, the differences between a guide RNA for an academic experiment where you're, elucidating some mechanism or doing some functional screening, versus identifying guides whose purpose are for introducing a therapeutic change, a therapeutic edit in humans. So it's very, very important that there is a very comprehensive and deep interrogation of the characteristics of that guide RNA with regards to on target as well as off target. And for this, at Intellia, we have developed a comprehensive workflow in which we apply both in silico and empirical approaches. We use genome-wide assays and next generation sequencing to verify, and identify what's the most active, the most precise, specific, clean target that does not have off-target effects. So I'll walk you on how we're doing this. So the first step, of course, is for a particular disease is to identify what's the gene, what's the type of mechanism you want to elicit. Do you want to do a knockout? Do you want to do an insertion? Do you want to do a correction? Then we select all of the potential guide RNAs that will target that gene. And, this is determined by the PAM, right? So these, you need to have a PAM as part of the specificity and site identification. So you generate a list of all of the potential guide RNAs, and then you apply a number of criteria to filter out some of the non-ideal targets. For example, you remove areas that, or guides that target domains that are present on other proteins, or that where there is some prevalent single nucleotide polymorphisms. You could also include guide RNAs that have cross homology across species. This is not absolutely necessary, but it really helps when you're trying to validate a target during preclinical development. Being able to use the same guide RNA in a mouse and in a non-human primate that you're planning to use in humans, it really helps with the pharmacology. And for this purpose, we use in silico bioinformatic tools, that you continually optimize with real data. So then you have your list of guide RNAs, and you use chemical synthesis to generate the test guide RNAs. And for that, we use solid phase synthesis. We use a 96-well plate format, and you can incorporate chemical modifications as desired by your platform. Then you use these guide RNAs in primary and secondary screens that you conduct on a cell type that will be relevant to the intended therapeutic use. So for us at Intellia, in vivo, we're going after hepatocytes in the liver. So we use primary human hepatocytes from various donors. And here we do 3 types of in vitro studies. First, we do in vitro primary screens at a single dose where you rank order the guides from the most active to the least active. You take the ones that are the most active, and then you do a secondary screen, which is an in vitro dose response. In both of these cases, we're looking at percentage editing at the on-target side, looking using NGS. And then you select based on the in vitro dose response, you select the most active guide RNAs, and then you conduct in vitro functional readouts where you're correlating the percentage editing to the consequence with regards to, for example, for a knockout, decrease in mRNA as well as decrease in protein expression. And you use this to select your most active guide RNAs for on-target activity. Then you need to look deeply into what's the potential for off-target activities, keeping in mind that as part of the design algorithm, you already took that into consideration. But in addition to those in silico predictions, we need to have experimental approaches to really pressure test what's the off-target profile of these lead candidates. And for that, we have a workflow that consists of an off-target discovery, and an off-target validation step. So what we do on the discovery end is we have the list of potential of targets from in silico algorithms, and then we do experimental approaches. There are a couple listed here. One that we like a lot is called site-seq, where we take chromatin, in our case from human, right? So, and you expose it to a Cas9 guide RNA ribonucleoprotein and digest the DNA, and then you use techniques to fish out all of the cleavage sites, and you can sequence them, and you end up with a list of all the potential loci where you saw a double strand break on target and off target. And you aggregate, you create a really long list of all the potential off-target site candidates. And then you generate a panel of NGS tools that allows you to interrogate each of those sites in using NGS analysis to evaluate whether, as a function of a dose response, you see off-target activity. And the goal is to select now the guide RNAs that have high on-target activity and at projected doses that are going to be therapeutically relevant, there is no detectable off-target cutting. And through this approach, you could identify a large number of what we call clean guides that will be suitable for clinical use. The approaches that I'm describing here is, these are well accepted approaches by regulatory agencies that support the clinical candidates that are moving into phase 1 clinical development. So, and the last part of analysis is for the on-target is to characterize any potential DNA structural variants that could come from that edit. And here you do short and long range amplicon sequencing, as well as direct visualization of the genome using unidirectional FISH that allows you to see whether there is any translocations or any other type of chromosomal rearrangements. All right, so now that we talked about the tools and how we come up with guides that are suitable for human use, is how do we apply them for generating therapeutic candidates. And, as I was telling you before, I'm going to tell you today about our in vivo pipeline and our in vivo approach. So for in vivo, CRISPR is the drug. So we are delivering Cas9 and the guide RNA into a human being for a therapeutic use. The organ that we have chosen is the liver, and the delivery vehicle is a lipid nanoparticle. I'll show you next why we made that choice, but the lipid nanoparticles are an effective mechanism for delivery of cargos, therapeutic cargos, to hepatocytes in the liver. So in a lipid nanoparticle, we co-formulate a Cas9 messenger RNA, as well as the guide RNA that's going to tell it where to introduce the edit. Once that this LNP is formulated, it's given to patients or to subjects by an IV infusion. The LNP ends up going to hepatocytes. They are taken up by a mechanism that is dependent on ApoE and LDL receptor. Once that's inside the endosomes, the lipid nanoparticle ionizable cationic lipids mediate endosomal escape. The mRNA gets translated in the cytosol into Cas protein. It binds to the guide RNA that was co-formulated with it. That ribonucleoprotein complex now migrates to the nucleus, The RNP scans the DNA, finds the PAM, finds the target site, opens up the DNA, and makes the double strand break. And then the DNA repair mechanism will elicit the edit, which for the first type of applications I will tell you is a knockout edit. So we're looking for indels. So why did we pick an LNP? Well, there are a number of key advantages that are shown on the right side. First is this is a clinically validated mode for delivery to hepatocytes. Second, they have a large cargo capacity, so we can fit a long mRNA coding for Cas along with the guide RNA. And if we wanted to have more than one guide RNA, for example, for an excision, we could do so. So we're not cargo size limited. And this is one of the many differences with a viral delivery approach, where it's a very limited cargo size. And in addition to that, the LNP there is no pre-existing immunity or de novo immunity. So all patients would be able to be treated with an LNP, and if necessary, redose with the same formulation. This is a no-no for AAV, as from the start, a significant percentage of patients will not qualify. They have pre-existing immunity against AAV, so they cannot be dosed. And then once that you're dosed, you develop immunity and cannot be redosed. Other important things on the LNP, they are biodegradable. So the lipid and the cargo is there long enough to elicit the edit, but then is gone, and that reduces the potential risks for lipid toxicity, for genotoxicity, and for an immune response against the Cas protein, because remember that the Cas protein is of bacterial origin. And then lastly, very practical for drug development is that this is scalable synthetic manufacturing. So you could make tons, you could make large quantities of guide RNAs at lower cost, higher efficiency in manufacturing as compared with viral vectors. And, as we have seen with, COVID vaccines and LMPs, right, they can be manufactured at scale to support very, very large patient populations. So now with the tools I just described, gene editing and delivery, we can now think about therapeutic strategies to remove, restore, or do a combination depending on the genetic change we want to elicit. So the first example I want to talk to you is about knockout. And our lead candidate, Intellia 2001, is in clinical development for a disease called transthyretin amyloidosis, or ATTR, and this is done in partnership with Regeneron. ATTR is a progressive disease, fatal disease, that's caused by the buildup of transthyretin, or TTR protein, in multiple organs. These could be wild-type protein as well as mutant protein that accumulates in nerves and in heart and in other parts of the body. And it can lead to two main manifestations, polyneuropathy and cardiomyopathy. There are an estimated 50,000 patients with hereditary ATTR worldwide, and between 200 and 500,000 patients with cardiomyopathy, wild-type ATTR. There are, over the last few years, there were a number of candidates that have been approved for the treatment of ATTR, but these are chronic treatments where patients need to take these drugs for the rest of their lives. So our strategy is to knock out the TTR gene in the liver, which is the source of systemic TTR. And by doing so, in a one and done approach, eradicate the production of TTR protein. And not only slow the progression of the disease, but depending on the extent of knockout, being able to reverse and potentially cure the disease. So this slide shows the validation of the approach in non-human primates. And this is a validation study for our clinical candidate. So what we've done here is we've given a single dose at 2 dose levels, shown in blue, of a lipid nanoparticle, again carrying the messenger RNA for Cas and a guide RNA that targets the TTR gene in hepatocytes for knockout. And as you could see, after that single dose, if you measure TTR protein in serum that comes from liver, you could see a very rapid decrease in the amount of TTR protein. And the levels drop by 4 weeks, you reach a nadir. And if you follow these animals for a year after a single dose, once that you reach that lower level, that nadir, that is stable. So once that you broke the gene in the liver, there is no way around it. There is no more protein being made. What's highlighted on the blue bar is what's based on the clinical data from other approaches for reducing TTR, what's considered therapeutically relevant. And the 2 levels, those levels that we're showing here, are way, within and beyond what's the therapeutic threshold. So if we are able to achieve these in patients, the expectation is that patients may have a one and done solution for TTR amyloidosis. So these serve as a validation of our first clinical candidate, NTLA-2001. We announced a few weeks ago that we have dosed the first patient in our global phase 1 study. So this study is a 2-part single ascending dose in part 1, and then an expansion cohort in part 2. The goal of the study is to evaluate safety, tolerability, PK, and PD for TTR knockdown. And the secondary objectives is to evaluate efficacy on clinical measures of neurologic function. We've selected patients with hereditary ATTR with polyneuropathy to do this phase 1 study, but we're planning to quickly move into all types of ATTR manifestations in the next clinical study. So, these are the achievements for this program. I want to also, highlight again that this is not only important for us, Intellia, as our first clinical candidate, but for the field, because this is the first truly systemic CRISPR candidate being evaluated in vivo. So now moving on to the next candidate, and this builds on the modularity of the liver editing approach. So the next program, which is called NTLA-2002, is in development for a disease called hereditary angioedema. Here again is another gene that's been made in the liver, secreted in serum, and important for this disease. The only thing we change from the previous program is the part of the guide RNA that determines the target sequence. The Cas is the same, the mRNA is the same, the lipid formulation is the same, and it builds on the infrastructure that we established for the TTR program. So everything that we've learned on how to manufacture, control the drug, all the preclinical development can be repurposed to the next program for a knockout target in the liver. And as shown in the next slide, if we're looking now at the validation, you could see the similarities with what I just showed you for TTR. So here we're knocking down a gene called KLKB1 that codes for a protein called prekallikrein. Prekallikrein plays a role in a disease, the hereditary angioedema. Due to time, I'm not going to go deeper into the pathophysiology, but the goal is to decrease the levels of kallikrein protein and kallikrein activity in the serum. And this is what we're measuring on the x-axis in non-human primates that were treated with a single dose of this LNP targeting KLKB1. And as compared to the control shown in gray, there are 3 dose levels. As shown before, you see a very rapid decline in the production of kallikrein, measured here as an activity assay. And once that you reach the nadir by 2 to 4 weeks, that is stable. Once that the gene was knocked out in the liver, it doesn't repair back to wild type. So you could achieve these very stable, profound levels of knockdown. Also shown as before in the light blue box is what's considered relevant based on pharmacological data of other agents in this disease. And the 3 dose levels that we tested in non-human primates that we expect are going to be therapeutically relevant and achievable in humans are all above and beyond that therapeutic threshold. So we expect them to be efficacious. And as with 2001, here we're also looking for these one and done, where patients will receive a single course of treatment with a in vivo gene editing, and when that gene is knocked out, the patients will have benefit for a very long time, potentially, a one and done cure. For NTLA-2001, we have guided that we will be submitting a IND or IND equivalent in the second half of next year. So I've been talking about durability, right, and the potential for one and done. I've shown you data, and let me go back here just to highlight that this is also a year after a single injection. And we've seen this multiple times, but 1 year is not a lifetime. So how can we find orthogonal ways of really pressure testing the durability of the edits? So to demonstrate persistence, we use a surgical model called partial hepatectomy. So what we do is we take animals, we introduce the edit, then we remove 2/3 of the liver, and we wait for that liver to undergo quick proliferation until it restores the liver size to the original organ size. And then the question is, what happened as those cells proliferated rapidly? Were those edits retained or were they lost? So as shown by the data captured in this slide, so here we have 2 groups of animals. We have control animals in teal and animals in which we knock out TTR in dark blue. So as you could see by the middle panel, after you do the edit, nothing happens in the control animals, but in the edited animals, we could get over 98% decrease in the expression of TTR. We then take those animals, we remove 2/3 of the liver, and we wait until the liver regenerates fully. Sorry. So, and then edits were maintained, were lost. And were edited and the loss of TTR, that it means that the hepatocytes that proliferated rapidly to regenerate the liver carried with them, so the progeny carried with them the edit. And this further supports our claims for long duration and this potential for one and done. We're able to edit hepatocytes in the liver that will contribute to the homeostasis of the liver and carry with them the therapeutic edit we introduced. So in the last 10 minutes or so, I'm going to tell you now about moving on from the knockout approach to a more complex type of edit called an insertion, where we're using an LNP to carry the gene editing machinery to make a double strand cut in the DNA in a precise location, and where we're going to introduce a therapeutic DNA. Here, the gene of interest or GOI. The way we're doing that today to deliver this template is through an adenoviral associated vector. And this is an efficient way of delivering DNA to the nucleus of cells. So where we're applying this, it's in a number of loss of function diseases. The first application is for Hemophilia B. This is a rare genetic disorder that's caused by missing, insufficient, or defective levels of a clotting factor called factor 9. And these patients, can have severe, spontaneous, serious bleeds. They require to be treated chronically with IV infusions of factor 9 replacement protein. So our approach is to insert the missing factor 9 gene in the liver so the liver can pump up into the serum the missing factor 9. So how we do this? We make a double strand break in intron 1 of the albumin gene. And we pick albumin because it's a highly expressed gene that is in the hepatocytes and that is secreted into the serum. So we make these a cut, and then we co-deliver a bidirectional insertion template having the human factor 9 in opposite directions. And the reason we do that is because in hepatocytes, they're non-proliferating, so homology directed repair is not really functional. So we're using non-homology enjoining to mediate and blunt insertion of the AAV insertion template. Because we cannot control the orientation of the transgene, we have this bidirectional construct. So whether it's in one direction or the opposite direction, in both cases, we're going to have a functional gene being expressed. So the way it works is that the albumin promoter will drive transcription because we're including a secretion signal from albumin. Our human factor 9 inserted is going to be actively and highly expressed and secreted into serum where we need it to be to control the clotting cascade. By using this approach in rodents, we can show using fish that about 50% of the mouse hepatocytes, they can stain positive for integration and expression of the factor 9 transgene. If now we look at mice for secretion of human factor 9 in serum, and here we're looking at 2 dose levels of the LNP AAV combination, we could see very dose-dependent and stable production of factor IX, looking for a 1 year after a single dose in these animals. What we found is that there are a number of ways of regulating the level of expression we want to achieve. I just show you those levels. The other one is the specific site for insertion within the intron 1 of albumin, the albumin gene. So here we're showing 10 different guide RNAs for insertion into that site. And as you could see, we have some guides that lead to very high levels of insertion and protein production in serum. Here is a matrix. So with this information now, we know that by changing the guide RNA we select, the dose of the template, and the dose of the LNP that carries the CRISPR machinery that will make the cut, we can titrate how much insertion do we get and how much factor do we end up producing and secreting into blood. We have now taken the data, the approach that we characterize in the mouse into the non-human primate, which is a more relevant species for human prediction. And as shown on the far right in this graph, comparing two different guide RNAs, one in teal, the other one in orange, we can range the expression of factor IX protein secreted in, activity in serum from normal levels to 10 times or greater the level of expression. So it's not that we're intending to overexpress factor IX, but it gives you an idea of the capacity of the system to really push expression of a gene if that was necessary for the therapeutic application you want to have. Here we're also comparing publicly disclosed data from 3 companies as a benchmark. The companies are using traditional AAV gene therapy, and we can, with our approach, we can match and much greater surpass the levels of expression. The next question is durability. So following the same thinking as I just described for knockout, now we evaluated whether the insertion introduced into a liver will be permanent if you push that liver to undergo very rapid cycles of proliferation. And as a comparator here, we use AAV, traditional AAV gene therapy, in which the construct inside the AAV has a promoter that will drive expression. And as a question, will that last? So as shown as the data included in this graph, where AAV expression shown in teal is rapidly lost after regeneration of the liver, here shown at 14 days and 45 days after partial hepatectomy, as compared to before the the basal levels that were achieved by gene therapy. The expression of the transgene, once that it is inserted into the genome, is stable. And what this means is that, again, the cells that proliferated rapidly to regenerate the liver carried with them and their progeny the inserted gene. By contrast, the AAV element is not integrated into the genome and remains as an episomal element. So as the cells proliferate, that episomal element gets lost. So this is a key differentiation for our insertion approach as compared to traditional AAV gene therapy strategies, and highlights the durability advantage that we provide. So, as shown you, I hope I provided enough data to show the modularity of the gene editing and delivery approaches for in vivo therapeutic editing in the liver for both knockout and insertion. One more example is that there are certain diseases, and one case here is alpha-1 antitrypsin deficiency, where you may want to do both. You may want to eradicate a mutant protein as well as to reintroduce a wild-type protein for expression. And this is what we've shown here in a mouse model of alpha-1 antitrypsin deficiency that express very high levels of a mutant protein called the PIZ gene. As shown by the graph, we first treated those animals with a lipid nanoparticle with a guide RNA that intended to knock down the expression of the mutant protein, shown in dark blue. And as you could see, there is a decrease and then eradication of the expression of that protein. A few weeks later, we came back with a different LNP that now made a double-strand break in the albumin locus and an AAV with no promoter, just to carry a DNA for insertion into that cut. And by doing that, we've seen insertion and expression of wild-type normal alpha-1 antitrypsin, driven now by the albumin promoter that led to expression and secretion of the normal AAT levels at therapeutically relevant levels. So we're very excited about the modularity of the platform that allows us to do knockouts, insertions, or a combination of both, depending on the disease. So the key takeaways, I think I covered them, but key messages is therapeutic gene editing requires a very careful and in-depth characterization of the guide's RNAs for therapeutic use, very clear and clean off-target profile. So that is absolutely important. Then to have robust tools that you can deploy depending on the disease to generate a differentiated and really robust pipeline. And the persistence that we have demonstrated for gene knockout and insertion, we believe is a truly differentiated approach of this technology that allows you to intervene early in disease. For example, for insertion, you could go in pediatric diseases, right? That, right now, given the unclear durability of AAV gene therapies, will make those approaches not durable. So with that, I want to close with a snapshot of the company and what to watch for next year, and looking forward to your questions. And I really thank you for your attention. So with that, I'm going to end the show and see you. Let's see. All right. Oh, and I see some questions here.
Yes, there's quite a number of questions, Laura.
All right, and I can see them. Yeah. So Chris Brown, is that the Chris Brown I know? Yes. So you can do that. So we do on and off targets. You do it in the intended cell types, but then as you know, LMPs, the majority of the dose goes to the liver. But there is a minority of the LNP that goes to, for example, spleen and adrenal glands. So you can, and we do look at potential editing in those cell types as needed. Let's see, let me go quickly through this. How are the LNPs targeted at the liver? Do they incorporate the Kanak ligand? No, so these LNPs in our case, we are using an endogenous mechanism. So the LNPs, once that they hit the serum, there is binding, there is opsonization of the LNP with serum proteins. One of them is called apolipoprotein E. It's very important for ApoE to bind to the LNP, and that's what's going to mediate its uptake by hepatocytes. So hepatocytes have LDL receptor, ApoE binds to the LDL receptor, and that's taken up into the cells. Potentially, yes, you could use GalNAc LNPs, but it is not necessary. How significant are the safety issues related to off-target activity? Right, so as I was telling you before, Jay. Hi, Jay. So we pick guides that do not exhibit off-target activity at at the intended therapeutic dose and a margin. So there is no that's part of the design, right? The design is to select guides that will not have this problem. So Lisa, how much knockout of TTR needs to be achieved for a therapeutic effect? So what we know from those who have come before us, and that is the work from Alnylam and Ionis using RNAi, whether it's an Onpattro, which is an LNP, or vutrisiran, which is GalNAc siRNA, or Tegsedi, which is an antisense. We know we need to be over 60% decrease in TTR to flip a trial positive, right? Now, the goal where we have seen therapeutic effect, right, on disease is to be above 85% TTR reduction. So, that's the goal. Chris Brown again. So if the LNPs are using ApoE and they're recognized by LDL receptors, but LDL is everywhere, how can it be that you don't get it anywhere? Well, there is a couple of great papers. I'll send them to you later, but these are particles at the end of the day. So you need to have fenestrated organs for these particles to make it and be retained on certain tissues. And there are other interactions that play a role. So heparan sulfate protoglycans, in the space of this also play a role in the liver for retention and then uptake into the liver. So we can talk more offline. So do you know if there is a limit for the number of guide RNAs? You can squeeze a whole bunch of them in there, but sorry, let me read it again. Do you know if there is a limit for the number of guide RNAs in case of multiplex mutagenesis using LNPs? So you can co-formulate several LNPs, and they're even with ribonucleoproteins, right? You could ex vivo by electroporation, you could put a whole bunch of guide RNAs, and all of them are going to cut the genome. In our experience, you want to have one edit at a time. And the reason is because if you're cutting the genome in multiple places, through this error-prone, rejoining of an edit of breaks, you could end up with a number of combinations that can lead to translocations and other type of chromosomal rearrangements. So ideally, do one at a time, or we're developing strategies that if we need to do an excision, is a precise excision. Do you expect any cytokine storm in the NHP study? So LNPs, this is well known, this is a class effect. They have a number of potential toxicities. They range from infusion site reactions to complement activation, cytokines, and liver tox. All of these are dose dependent, are monitorable, and are reversible. And the goal is to be able to have your therapeutic edit at doses that do not cause any toxicity. And this is what you do through your GLP toxicology studies, where you determine a therapeutic window And for the clinic, you try to be within that therapeutic window. But if you were to see any of these effects, like have been seen in the clinic by others, they're monitorable, reversible, and there is pre-medication with dexamethasone and with H1, H2 inhibitors that are given to patients to reduce the risk of any toxicity. Okay, thank you. Sorry, I'm reading. I'm looking at the time, so I'm trying to pick a few. And you guys are welcome to email me later if you want to chat. But we're using chemical modifications in our guide RNAs, yes, at the ends and elsewhere. And we make sure that these modifications do not impact. In fact, they help with increases in potency and do not cause any trouble for off targets. Is the scaffold region of the guide RNA always the same? Yes, it is always the same. At least, it changes depending on the system that you're using, but for a particular system, it's the same. Eliza, to understand correctly, in case of a factor 9 insertion in hepatocytes, are the hybrids, these hybrids you use to transfect the cells, LNPs containing an AAV? No, they are separate. LNPs carry the machinery to make the edit, and the AAV given separately as an AAV carries the promoterless template for insertion. Did you control whether the insertion is in hepatocytes? Yes. Sorry, I'm just... what's the mechanism of the guide RNA and Cas9 entering the nucleus? You have a nuclear localization signal. Chris Brown, my God, you're like too many questions today. Are antibodies made to the Cas9 protein presented on the cell surface of hepatocytes, or would this limit how many times you can go to edit the genome? No. Guide RNA, because the Cas9 is produced inside the cells, even if you were to have a humoral response to antibodies against Cas9, they don't meet because that's on the serum and the Cas9 is inside the cells. And then last question, Yun, how do you ensure that the hepatocyte being transfected by LNP and AAV are the same one? Great question. So at the doses we're using, most of the hepatocytes get an LNP. And as you know, AAV transduction is not that great. So, but whichever hepatocyte gets an AAV already has an LNP. I think I cover everything. Oh, one more. All right, enough. Reach out later.
Thank you very much, Laura. That was a fantastic presentation. Thank you very much for doing that for us.
Sure.
I'd just like to finish it off here because Laura has some other commitments now. Just to remind people that the next OTS webinar will be in January with Annabelle Biskans. And then in February, we will have Dr. Stanley Crook talking to us about N-Lauren. This is about the N=1 therapeutics, oligonucleotide therapeutics. Thank you very much, everyone. As usual, this has been recorded and it will be available from the website soon. Bye. Have a nice day.
Thank you.